we first met about a little over 5 years ago when I was interviewing for the job. And I remember at that time, Lilly had a drug that had just failed. And I remember kind of being new and a little bit naive to the whole field and the science of Alzheimer's. So I asked her, I said, okay, this Lilly drug failed and this plaques and tangles theory. And I remember you saying to me, really and truly, this is over 5 years ago, hey, the trial I'm really watching is the Biogen trial because of the PET scan. And this is the trial to watch, right? And here it is 5 years later and prophetic. And you knew it because it was based on the science, Howard. Let me come back to Howard. This FDA approval, I think some called it a provisional approval, but it's really an FDA accelerated approval. Can you just walk people through what does that mean? And there's in the media, there's been a little bit of controversy around this, and I don't want to get hung up on the controversy because we have a very distinct view here. But if you could just simply walk through what did the FDA say in approving this, because they were pretty open about how and why they approved it, and we don't need to get into some big controversy, but where there's a little bit of a confusion or rub, if you will. So.
Yeah, I think the thing that was driving some of the controversy is the ambiguity in the data, and particularly in the clinical data. One trial was positive on the clinical data, and another trial was essentially negative. And usually the FDA approves drugs on the basis of what they call substantial evidence of efficacy in 2 phase 3 trials. So the FDA was faced with a very difficult decision. They could have done one of 3 things. They could have rejected the drug and told Biogen to do another phase 3 trial, which would have cost maybe $500 million and 3 to 5 years of delay in patient access. They could have approved the drug without any restrictions, which wasn't warranted because of the ambiguity in the science, and particularly, again, in the clinical data of efficacy. Or they could have done this accelerated approval, which is sort of a conditional approval, as you mentioned, Mark, where the drug is enabled to have patient access and marketed, but it's required that the company do another phase 3, what we call a phase 4 clinical trial, since it's post-approval, and to show that the drug actually works in the clinic to slow the rate of decline of patient symptoms and function in a meaningful way. And if it doesn't, then the FDA can pull the drug from the market. Now, the last thing I'll say, which is again really critical, is that the accelerated approval process is very much dependent on the biomarker. And again, the biomarker here was the PET scan that ADDF helped to develop more than 20 years ago. The point is that what the FDA said, which is quite incredible, is they equated the PET scan with being a surrogate marker for Alzheimer's disease. Now, if you want to think of surrogate markers, cholesterol is a surrogate marker for heart disease. So that's the level at which this biomarker was taken as data, because the pharmacology of the drug worked. It did what it was supposed to do. It removed the plaques. And the FDA took one step further and said that it was reasonable to think that removing the plaques resulted in clinical improvement, which I think is a place where there is some controversy about whether that statement is warranted, but it really formed the basis for this conditional approval.
Very helpful, Howard. And I want to remind folks, I've got one other basic question to ask Howard here, and then we're going to open it up to the Q&A from the audience. So please use the chat feature. Just click on it, put your question in there, and forward it through, and we're going to do our best to answer everybody's questions here. Although, Howard, before we go on to the last question, which is, what's your and ADDF's take? What does this mean for the field writ large at this point? I have one question for you, just based on that and based on the accelerated approval. Howard, you're true north. You keep a small concierge practice of patients, and you always say your true north is your patients. This is all about what can we do for the patients and the families. Howard, and I understand the drug for short is called Adu, A-D-U. Howard, in your practice, given what you've seen as the approval from the FDA here, how would you think about it in your practice?
Yeah, that's a great question. I certainly think that I have an obligation to do what's called shared decision-making with my patients. My job as a physician is to give my patients information, and then together we make a decision about care. And in this case, I would present the evidence for and against efficacy here. There are safety issues here that need to be monitored. There's a burden of tolerability here. This is an IV infusion once a month that requires a lot of imaging, and about 40% of people will have abnormal imaging. So to keep my answer short, I would discuss the pros and cons of this therapy and expectations of what it would look like while they're taking it, because patients are going to continue to decline, and we're really not going to know if someone is a responder. And then, together we would decide whether or not to take the drug. I would not say you shouldn't take the drug, because I think patients need to help. They need help in making that decision, and if they want to take the chance of doing it and the burden of doing it, then I think they are entitled to do that, particularly since it's FDA approved. But I think we have to manage expectations about efficacy, and I think that's where the drug potentially could run into trouble, because long-term compliance and persistency with taking this drug over many, many months, I think could decline as patients are burdened with the infusions and so on, and aren't finding themselves getting better, which is an unrealistic expectation, because patients actually won't get better in all likelihood. They'll get worse, but at a potentially a slower rate of getting worse that we won't be able to measure in the clinic.
Howard, I saw one of your interviews in the run-up to the decision, and I think you were reminding a reporter that, these treatments, it is not about saving life, it's about improving quality of life. Right. And I thought that was very, very well said, and a good kind of barometer for all of us. Howard, that leads into, the last basic question here. It's great because we got a ton of questions coming in from the audience. But Howard, okay, let's look at the landscape of clinical trials. I've heard you say that this is the beginning of the modern era of clinical trials. We have blood tests now, we have PET scans. Adu just got this accelerated approval, but how do you really look at the broader clinical trial landscape right now?
Yeah, as you know, Mark, I've been at this about over 40 years, both on the research and care side. And as I've said many times, I've never been more excited. You've said many times that more than half of the drugs in development now are innovative, novel approaches to therapy. They're non-amyloid, non-tau approaches. They're against the inflammation in the brain and the metabolic disorders in the brain, epigenetic disturbances that are common in neurodegeneration. And what I think is that regardless of whether aducanumab was approved, we have so much momentum now in clinical trials. ADDF alone is supporting 35 clinical trials in one space or another around the world, of 35 different drugs. So we have momentum, and I think the approval of aducanumab, or the conditional approval, adds weight to our portfolio and our approach. But regardless of that, we're going to keep moving forward, and we have a lot of momentum now, and I'm really excited about the future. And I think that in the near future, we might potentially have more approvals coming down the pike. Again, as I mentioned, drugs that attack inflammation in the brain and so on. So I'm really excited. I'm glad we got a conditional approval, that there's a hint that the drug works, but it's only going to be an incremental benefit. We're going to need combination therapy. We have biomarkers now, so we can practice precision medicine and run these trials in a more precise way on targets that patients have with these novel biomarkers. And this is, as I mentioned, just the beginning of, I really do believe it is the beginning of the modern era of Alzheimer's clinical development and new drugs.
Terrific, Howard, terrific. And perhaps you and I will come back and visit that, but there's a ton of questions here. So Howard, let me start giving a feel. There's several people asking about, and I think you touched on this a little bit, but Ken Moore, the FDA wrote what is called a broad label, right? So what is your view on that? And can you explain that a little bit to folks if they don't?
The problem with the FDA label is that there are no guidelines by which physicians...
And Howard, can you just, when we say broad label or the breadth of the label, what does that mean for folks who may not?
Right. So the FDA regulates how drugs can be marketed, and that's called the label. And so there's already a document out there, which is the labeling document. And two things that the FDA did not do in the label, which I think is critically important and actually a problem, is that they did not require a PET amyloid scan or the amyloid blood test, or even a spinal tap, to identify patients who have amyloid in their brain and would be candidates for this therapy. That was one of the problems in the clinical trials, and one of the advantages that the PET scan brought to the world is that we realized that 30% of people that were being entered into clinical trials for anti-amyloid trials didn't have amyloid in their brain. The clinicians got it wrong. The Medicare IDEA study showed that half the time, so-called dementia experts got it wrong. And so we don't want to be treating people that don't have amyloid in their brain who haven't gotten the test, and the label doesn't require a test. And then the second thing is, this trial was done in a very narrow group of patients with mild symptoms. But the label says, basically, you can treat anybody. Imagine somebody comes in with a memory complaint. Now that's going to be a second level of regulation, which will probably be done by Medicare and by the Medicare managed care organizations that will create what we call health technology assessments from the pharmacy and therapeutics committees at the Medicare Advantage plans. They'll create guidelines. They'll probably create prior authorization procedures that will require the PET scan for treatment, and then have stopping rules if patients don't seem to be responding. So the payers now really play a critical role to fill the gaps in what the labeling that the FDA created did not do.
Got it. Totally got it. Hey, Harry, there's a question from our good friend Susan Dickinson in here. You know, first of all, there are several different types of dementias, right? But you know, in your opinion, what does the use of this accelerated approval pathway by the FDA signal for rare diseases such as FTD?
Yeah, I think a few years ago, the FDA put out new guidelines for the definition of Alzheimer's disease, and it was biomarker based. So that means it's kind of like cholesterol. You know, people can have high cholesterol, they can have hypercholesterolemia, they can go on Lipitor or statins, even though they haven't had a heart attack. And I think that analogy applies here, that the FDA basically recognized 4 stages of Alzheimer's disease, and the first 2 were biomarker driven. And they would approve drugs, even people who, they said they might approve drugs for people who were asymptomatic that had biomarker evidence of the disease. And we know the disease starts 20 years before people get symptoms. I think the same thing's emerging with the frontotemporal dementias. There's a lot of effort, and part of that is being driven by our wonderful partnership with the Association for Frontotemporal Dementia and Gates Ventures and other philanthropists through the Diagnostics Accelerator to develop biomarkers, hopefully blood tests for frontotemporal dementias. And I think that's the key. That's what enabled the Biogen study and the approval, the biomarkers, the PET amyloid scan that ADDF developed. And that's what's going to happen. That's what's happening now in the FTD types of dementia, that once we get the biomarkers, it's really going to up our game in clinical development. We're really going to be able to do better clinical trials for the frontotemporal dementias, and I think it'll accelerate drug development. So we're just on the cusp now, I think, of having those blood tests and imaging tests that are needed to make the FTD trials more precise. They're very difficult because the FTDs are relatively uncommon, and identifying the patients is one of the issues. So, but I'm optimistic. I think FTD is going to move forward much more rapidly now, and the FDA will recognize these biomarker stages of FTD. And I know there are trials already going on by industry in this space using biomarkers. So it's an exciting time for FTD also.
Our good friend Carolyn Weiner has a wonderful question, or a very good question. You know, in the preliminary data, the side effects like brain swelling were far more common in the APOE4 carriers. You know, did the final data confirm any of that, or what more can you say on that?
Yeah, it is looking like that. I mean, all the trials these days look at APOE4 genotype and the way that people respond differently if you're APOE4 positive or not. And it is true that the APOE4 patients have more of that side effect. So that's a consideration. And again, when we talk to patients in the clinic, I think what this is going to do is make ApoE4 testing, ApoE testing, getting people's genotype more common, because when we discuss with patients the risks and benefits of therapy, we're going to want to know their ApoE4 genotype so that we can give people a better estimate of what their risk might be, as Carolyn mentioned. And so I do think it's an important issue in that shared decision-making discussion that I mentioned to you that physicians will need to have with patients. If a patient is ApoE4 positive, they'll be at greater risk of the side effects. But I have to say that while the side effects are pretty common and they're detected by MRI, and MRI monitoring will be required over the course of therapy, most of the time, almost all the time, these side effects are not progressive. They're usually self-limited. The symptoms are headache and confusion and dizziness, and it's temporary. And the brain imaging, edema and hemorrhage, these microhemorrhages, usually resolve themselves. So I don't think the side effects per se are rate limiting, even in the ApoE4 patients. But we really don't know, because we don't have that last phase 3 trial to demonstrate the real parameters around risks and benefits.
Got it. Hey, our wonderful Board of Overseer member, Charlie Cangro, has a question that I think, I've seen a couple of different things on this, but you'll be able to land it really easy. Aducanumab, in the trial where it did show some positive effect, was that for the earlier stage, the MCI? Was that for the later stage? And was there any data around, the type of patient this helps, has the possibility of helping the best?
Well, that's the difference between the label, what FDA approved, and what was done in the clinical trial. So in the clinical trial, basically patients that were tested were early mild patients. There were no patients with moderate to severe disease who were tested, and there were no patients that had really, really early disease with just a few memory issues. And so particularly on the moderate to severe side, I imagine, the label leaves it open that physicians could treat people with moderate to severe disease. But as we all know, this is a very expensive therapy. It's burdensome, as we've discussed. And it's hard for me to imagine that I would recommend to my families that people with moderate to severe disease, sometimes even bedridden or requiring 24-hour care, that I would see the benefits of treating those patients to slow the rate of decline, really outweighing the risks and the burden of getting that therapy, and the cost, I should say, because as you know, the cost here is about $54,000 a year, and that doesn't include some of the copay costs and other costs that patients themselves might have to bear. So I think that in practice, the moderate to severe patients probably won't be treated, and the guidelines might not even include those patients, even though the FDA didn't address that issue in the label.
Interesting that we bring up cost, because we have several questions about that. And I'm going to ask our staff that's kind of moderating all the participants on, George Reimburse, who's the founder of the wonderful organization Us Against Alzheimer's. Howard, he has a question that, I already know your answer to, but I'm going to ask an answer to. But I'm also going to ask if we could open up George's line, because there are questions around cost, and there are questions around, who can get into trials. And I think, as many of you know, with the ADDF, we are solely on funding science. We don't do awareness or advocacy or patient care. And there are wonderful organizations that are our partners that do those things out there. One of the very best is George Reimburse's Us Against Alzheimer's, and what they do inside the Beltway on the advocacy side is just amazing. So Howard, George's question, and then George, and we're going to make you live to answer a question, but George's question is, does Medicare reimburse for the PET scan?
We do not have reimbursement for the PET scan. I hope that with the advent of aducanumab on the market, that they'll reconsider that. And I know they'll be lobbying, not from ADDF, although we might support that lobbying, to get reimbursement for the PET scan, because I think it's absolutely essential that people have that biomarker positivity before they get treated, because the risk of treating so many people that don't even have amyloid in their brain, which is the target of this drug, is very serious. Now, we do have a blood test, right? So ADDF's been investing in this company, C2N, and they got clear approval in 49 states now, which means they can market and roll out this test. And I've actually prescribed the blood test for several patients in different states. So it may be that while the brain scan costs $5,000, the blood test is only $1,000 or something like that. And maybe what can happen is that CMS or the payers, Medicare Advantage, could approve the blood test as being good enough. I don't know. But I think the clinical practice guidelines that doctors will use to administer this drug have to include the PET scan. And that PET scan has to be reimbursed, or otherwise the access to this drug is going to be very, very limited. Most people cannot afford to pay $5,000. Now with a lot of volume, maybe the price of the test will come down, but there'll be a lot of people that will not have access to the drug because of the price of the PET scan.
And that slides into also the price of the drug. And George, I know you have the ability to chat. So first of all, George, what a moment. And also I have to say about George, he has been saying for the last 2 years from any podium he's been given, hey, new treatments are coming. We're going to have to start figuring out, accessibility as these treatments come. So George, you've been another one with great foresight, like the lawyers, like Howard. George, perhaps I suspect from where you sit, why don't you comment for a second on your view of this moment? And then could you talk about from, you are the leader in the Alzheimer's space around things like accessibility to clinical trials. So could you give us your comments on that?
Well, Howard has hit the nail on the head. It is important, indeed critical, that we persuade Medicare to change its position on the reimbursement for PET scans. And I am reasonably confident that there will be a solution short term and then longer term very, very quickly on that score. But we'll see, because Medicare itself is a complicated agency with a lot of decision processes and deeply concerned about the overall cost to Medicare system of reimbursing for the diagnostics and for the drugs. And Howard is right that I think in the medium term, that blood tests will begin to overcome this problem of cost and accessibility of the diagnostic process. But there's no question that patients are deeply concerned about what their out-of-pocket costs are going to be. Now, for many Medicare beneficiaries, whether they be on Medicaid, which thus covers all of the drug costs, or for those who are on fee-for-service but have a supplemental benefit policy through AARP and United Healthcare, most of the out-of-pocket costs will be covered. Still some, but most covered. The real troubling element is people on fee-for-service who don't have a supplemental benefit. And for them, they'll be paying 20% of the drug costs, and that's roughly $10,000 a year, which for many, many families is just simply out of reach. So in addition to all the burdens that Howard described of getting on the drug, infusions every month, not seeing progress, we also have these costs, which for many low middle income populations is simply going to mean that the drug is not accessible. And that's intolerable because whatever the benefits of this drug might be, they should be available to everyone to make the decision. And so, yes, we will be working very hard on trying to persuade Medicare and other insurers to indeed make sure that the out-of-pocket costs for low-income populations are reduced really quite significantly.
And if I could add to that, Mark, I was at the committee and testified at the Medicare committee that denied payment for the PET scan. And one of the main criticisms at the time was that even if you get the diagnosis, there's nothing you can do. And I got very angry about that because, obviously I've been taking care of people for over 40 years. So telling me that there's nothing I can do for people is kind of ridiculous. But what they really meant was in the typical medical model, there were no treatments. Now we have a treatment. So when we go back to the committee, it's going to be more an issue of cost effectiveness. And even though Biogen is saying this, they're pricing this at $54,000 to $55,000 a year, the Institute for Clinical Effectiveness Research, which is sort of the standard of cost effectiveness research in the United States, and testified at that committee meeting of Medicare price reimbursement for the test a few years ago. They estimate that the cost effective price of aducanumab is somewhere between $2,500 and $8,500 a year. So there's a huge gap between what the analytics say, what the cost effectiveness price is, and what Biogen is going to price this at. And ultimately, it's not even so much the pricing, it's the budget impact on the Medicare budget. Because you can have a $1 million test that affects 12 people in the country and there's not a lot of budget impact. But the problem here is you've got an expensive drug at $54,000 a year and millions of people that could have access to that. The budget impact on the Medicare pharmacy budget is huge. And I do want to say one more thing, if I may insert here, is that ADDF really contributed to the intellectual property that resulted in this test, the Amyloid test, that enabled Biogen to get to this approval. And through our venture philanthropy, I just want to say that I'm very proud that we do get a percent of royalties that the University of Pennsylvania receives. And this is said in the spirit of transparency, but it confirms our model, right? Because the money that we may get going forward and that we have gotten in the past, both in the clinic and for research purposes on this test, will be plowed back into more research. And the reason that we funded this test is exactly 20 years ago, about 12, 18 months into the start of the foundation. The reason that we funded this thing at Penn when no one else will is exactly the reason that Biogen used it to get the approval, which is based on this surrogate marker.
So, which is great. So thank you. And also to George, thank you for your leadership and ongoing partnership. Howard, I want to turn this back to a little bit of just kind of the scientific landscape here. You know, Howard, I also remember when I started, you said to me, hey, there's been a lot about amyloid out there and most of it's failed and you're keeping your eyes on this Biogen test. But you also said that, 8, 9 years ago, as you know, it's interesting you said about our venture philanthropy model, right? 10 years ago, ADDF was funding things around amyloid, plaques and tangles, and then Big Pharma came in, and so it was time for us to pivot to be out. And you and ADDF took an 8 to 9 year run at looking where the light's not shining. And you've described it as the biology of aging. That's where all, as you and I are here today, both of us are aging and we age differently based on a series of different factors and quite frankly, pathways. And that this is now about an opening up of pathways. So there's a couple of questions here kind of to step folks through. This Adu drug is about amyloid. There's also tau. There's also genetics. There's also inflammation. There's also vascular. Give us the perspective, and even with Adu on amyloid, right? We know Lilly still has an amyloid phase 2 going to phase 3 that's out there. So can kind of, is that all cluttered or does that all make sense to you? And how do we look at it?
Well, I'll try to be brief, Mark.
Yeah, this one tease out a little bit, Howard.
Okay. I have a great vignette, which I probably don't have time to tell, but we started a company out of the Oklahoma Medical Research Foundation in the year 2000.
Yeah, you got to make this one shorter, Howard.
We had the first anti-amyloid pill, basically, in the clinic, showing that a beta secretase inhibitor could lower the levels of beta amyloid in the blood. It was the first drug that showed any efficacy. But as you said, as the research on anti-amyloid therapies progressed and the role of Big Pharma started coming in and Big Pharma started spending billions of dollars on clinical development, it was time for the philanthropy to step back. So it wasn't whether or not we believed in amyloid hypothesis. It was really that the time of affordable, low-cost innovation had moved on to phase 2 and phase 3 studies that cost literally hundreds of millions of dollars. And so that was the right role. Let's take the case of amyloid and aging. So all of our proteins in our bodies misfold. Proteins have to be in a certain three-dimensional configuration to work, to function. The analogy I like to make is, taking an egg that's going to become a chicken and heating it up on a frying pan, and it becomes a scrambled egg. That's misfolded proteins. The misfolding is occurring because of heat. That misfolding is happening in every one of our cells every single day. Through evolution, if we didn't have ways to manage that misfolding, we would die. Our cells would die. So that process of managing misfolded proteins, which happens every day in every cell on multiple proteins, is called proteostasis or autophagy. It's been a difficult target, but the point is that in the plaques, there's not just amyloid. There's many, many other misfolded proteins that are kind of sequestered in these extracellular plaques. The body sort of just congregates them into the plaques. And so we know that these plaques have many misfolded proteins. And that's why I believe the anti-amyloid approach is incremental. It works a little bit. But as you mentioned, Mark, there are many other misfolded proteins in the aging brain. Many, TDP43, tau, alpha-synuclein. I could go on, with big words, but there are many. So what we need is a pleiotropic, parsimonious, again, big words, but you need a pleiotropic, parsimonious approach to these many misfolded proteins if we're going to have a big effect. And that's what we're focused on now. We're not focused on one antibody or a cocktail of two antibodies. We're focused on the process that goes abnormal or gets overwhelmed with aging that leads to the deposition of these proteins in the plaques. And that's something we're very excited about. And it's starting to catch on in industry. There's a few programs in industry that we're aware of that we're trying to invest in now. And as you mentioned, Mark, that's just one pathway in the biology of aging that we're addressing. Inflammation is considered one of the hallmarks of aging. As our bodies break down, there's an inflammatory process. The inflammatory process is normally to remove these damaged proteins. It's a protective process, but with aging, it goes awry. Okay, it becomes overstimulated. And we need to parse that down in the aging brain because we know, for example, that people can have amyloid in their brain and other misfolded proteins. But if they don't have an inflammatory response to that, to those deposits, they actually don't progress very much or at all. And there was a clinical trial at the University of Southampton in the UK that we supported of a major anti-inflammatory drug called Enbrel to see if we could identify people who have inflammation in the brain. And the problem there is we don't have the best biomarkers yet to identify those people. So that's something that we're invested in because we want to find those people that have the amyloid plaques and have the inflammation around those plaques because those are the people that progress. And so what we want to do is treat those people maybe with aducanumab, maybe with an anti-amyloid drug and an anti-neuroinflammatory drug. So we're building this incremental combination therapy strategy, the same way that, if you read Siddhartha Mukherjee's book on the Emperor of All Maladies, he describes how back in the 40s, they started with one toxin and then they built on with another toxin because to kill the cancer cell, you've got to hit multiple pathways. And cancer is a disease of aging. So we're learning a lot from cancer. So, I'll be brief. I know I'm supposed to stop here.
No. This is good, Howard.
Okay, but those are just two processes of aging that I described. The last thing I'll say, actually, is we're very excited about neuroprotection. And we're going to hear a readout this summer on what I hope will be a major advance, a program we've been supporting for 20 years. Frank Longo, who's now the chairman of neurology and neurosciences at Stanford. Because if you have these many different ways of neuronal injury, and it's like whack-a-mole, you want to try to hit the autophagy, hit the inflammation. But imagine there was a drug that, regardless of the cause of the injury, was protective of the cell. That's called neuroprotection. And we're in phase 2. We're gathering the data now on the results of the clinical trials. We're going to hear probably in July at the AAIC meeting on whether that drug worked. And that neuroprotective drug that we're supporting with Frank Longo through a company that we helped him create called Pharmatrophix. If that drug works, I will humbly say that it really could be a very big breakthrough for our patients with Alzheimer's and other neurodegenerative disorders.
So Howard, that's great. And there's a couple of ways I want to take this. So let me try. If we get often too wonky, I'll pull us back. You talk about the inflammation side. There's also vascular that plays a role. There's also genetics that plays a role. Can you give a comparable feel for those two pathways?
Sure. Well, we started funding a program at Rockefeller University several years ago that's resulted in a company in San Francisco today called Fillit. And it's really interesting because what happens is that there are proteins in the blood that are clotting proteins. One of them is called fibrinogen. And it binds amyloid very, very tightly. And when it binds amyloid in the blood vessel, by the way, there's amyloid in the blood, obviously, that's why we can have a blood test. And so when there's vascular damage of any kind, you get this clotting mechanism with fibrinogen activated. And the fibrinogen binds amyloid very tightly, forms a complex that's very hard for the body to remove and results in inflammation. And that inflammation is thought to open up the blood-brain barrier and lead to neurodegeneration. So what Fillit is doing is that they have antibodies that inhibit that interaction, very specific antibodies that inhibit that interaction between fibrinogen and certain fibrin products and beta amyloid. And they've shown in animal models that this theory works, that these antibodies work, and they're on the cusp of entering into the clinic with this. So that's just one example of how we're approaching the vascular component, because vascular disease is really common. Practically all of us have vascular disease. Over 85% of people over 65 have vascular disease, right? And that's why diabetes and hypertension and atherosclerosis are risk factors for Alzheimer's disease. We've got to pull these two things together. You have a neurodegenerative disorder with plaques in the brain, and you've got this systemic illnesses that are risk factors. How do we put that together and explain why is hypertension a risk factor? And it may be through many mechanisms, but this fibrinogen mechanism may be one of them. And it also gives us rationale why we're repurposing antihypertensive drugs with neuroprotective properties and diabetes drugs with neuroprotective properties for Alzheimer's disease. I mean, I'm really excited about these approaches. We're going to hear about the results of the candesartan, which is an antihypertensive drug, over the summer. There's a lot of evidence that these drugs can be neuroprotective. We've already heard about the neuroprotective properties of the diabetes drugs, GLP-1 agonists like liraglutide, and Novo Nordisk is already along the way of doing a diabetes drug called semaglutide for Alzheimer's disease. There's a big clinical trial going on there. You asked me about epigenetics. One of the main ways that cancer is treated is by identifying genetic abnormalities in the cancer cell, and then using what are called epigenetic drugs to try to fix these epigenetic marks on the DNA and the histones, which are the wrappings of the DNA that regulate how DNA is read and written and repaired. And these are epigenetic drugs. There's many epigenetic drugs on the market today for cancer that attempt to fix the epigenetic. Now, these are not mutations, but they're marks on the genes that allow the genes to be regulated and expressed under various circumstances. It's the reason why kidney cells don't become neurons, or neurons don't express kidney genes. And so what we're doing is we're surveying the field of epigenetics to understand, because there's data that epigenetic abnormalities also contribute to neurodegeneration. And we invested in a company called Rodan Therapeutics that was spun, I believe, if I remember right, out of MIT, and was supported by institutional investors that have an epigenetic drug. And we're also supporting a company out of Madrid on the Madrid exchange called Horizon that's in clinical trials now with an epigenetic drug. And we're looking for other opportunities strategically to address this, because epigenetics is a critical part of aging. The epigenetic abnormalities are probably the reason why cancer is a disease of aging, and the same rationale. And the only difference, and I'll end here, is the difference between how the biology of aging affects cancer cells and results in tumors and the neurons is that cancer cells are dividing cells. And when you get abnormalities in cancer cells, like epigenetic abnormalities in cancer cells with aging, you get abnormal proliferation because those cells divide normally, right? They're dividing cells, and the division properties get out of whack. But neurons are not dividing cells. You don't want those circuits to be dividing. You want them laid down. Okay, I remember how I tie my shoes. You want that for 70, 80 years. You don't want those cells to die, and you don't want them proliferating because it'll muck up the circuits. So when neurons get epigenetic abnormalities, they die. They kill themselves to get out of the way. And so we want to take the same processes and understand them. You know, cancer's way advanced on us in this, and learn from that and get in strategically into that field. So I won't say more, but we're having.
Right, but Howard, I have one other short one to this, and then there's going to be a bit of a wind up here as you know, we got about another 10 minutes or so. But Howard, what about you know, when you get into this, looking at this genetically, you know, a vaccine for Alzheimer's?
Well, the monoclonal antibody is a vaccine. It's a passive vaccine. So there's two kinds of vaccines. There's an active vaccine and a passive vaccine. Active vaccines, example, are tetanus, diphtheria. You give the virus to the person actively develops immunity to the virus, and it's permanent. Okay, and there's an article today, a really interesting article today, about how we get permanent immunization from active infection because the T cells are involved and even the memory B cells. And this has been known for decades, so it's not new. So that's active vaccination. And then there's passive vaccination, which is the anti-serum approach that was used back in 1900, you know, when Pasteur and some of the others were developing vaccines, active vaccines. They also tried passive vaccines from horse anti-serum and animals like that. And they worked. They just had really bad side effects because people didn't like getting horse blood, and they died sometimes from getting horse blood. So what aducanumab is, a passive vaccine. There are companies out there like AC Immune and even Elan Pharmaceuticals, which is no longer active, that had an active vaccine. They actually injected forms of beta amyloid into people's muscles so that people would develop a normal immunity. I don't know that that's really being actively pursued or widely pursued around industry right now. The problem is side effects. You know, if you get side effects.
But we are looking at gene therapy.
I think gene therapy, you know, we're supporting some gene therapy programs at ADDF trying to fix the ApoE4 genotype. And I think gene therapy has a lot of promise for Alzheimer's disease.
Yeah. So, you know, Howard, I want to kind of take this back to the beginning of both ADDF and also, you know, we're here today because this approval of aducanumab, and just to give kind of a holistic feel here. I want to start with, you know, I'm going to go back 23 years in the Lauder family founding ADDF with you at the helm to only fund translational research, to go from bench to bedside, to get 20 years out ahead of this, because the folks at the NIH said at the time, hey, we got plenty of money for basic science, but nobody's taking, you know, we know enough about the brain then to start taking some shots on goal. And I also want to give to the boards of ADDF a lot of credit, because 10 plus years ago, when Howard came to them and said, look, there's this plaques and tangles amyloid theory, big pharma is coming into it. I kind of want to turn to where the light's not shining and look at this biology of aging, and they approved the strategy, and with this real sense of it's about multiple shots on goal. There are 6 or 7 of these different pathways outside of or complementary to the amyloid hypothesis. Go after that. And I also want to point out that another genius of the founding of ADDF, which was the Lauders, and Howard was there then, and folks like Alan Green there then, who said, you know, this will be a venture philanthropy. We're not going to give out grants per se. We're going to have contracts with academics and with biotechs, so that if there was any success that was generated, any equity success that was generated by these companies or institutions, there would be a payout to ADDF that we would put directly back into the science again. I bring that up because there was a question about, is there any financial residual that we're getting from the Biogen drug? And the answer to that is no, because again, 7-8 years ago, we stopped investing in the amyloid hypothesis. We'll get there, Howard. I promise you we'll get there. Trust me, it's all coming to you, Howard. So don't worry. We'll get there. We don't. But we have a very, very wide and deep, and you heard Howard comment on it. We are the world's largest funders of clinical trials. 35 different clinical trials that we are actively funding now, with more coming on. And the big piece I want to hit here with all of that is what Howard has said. Great that Adu has gotten this approval from the FDA. It is an incremental step that opens up this modern era of clinical trials, which it is about prevention and combined therapies. And this is what everyone who's on here in our lifetime now, this is beginning to happen. And those who are younger than us and the generation after that, they're gonna benefit from these very moments that we're in. So Howard, I'm gonna come back a little full circle. Given Adu has gotten approved, given what you see now across the landscape, final thoughts. But I want to do final thoughts with, and you've mentioned one or two of them. Are you seeing things that you think come next year?
Yeah, well, absolutely, Mark. And just let me comment.
Please.
As I mentioned, the critical thing that enabled the Biden study and the approval, as recognized by the FDA, was the Amyloid brain scan, which ADDF invested in, and we will get royalties on the utilization of that brain
Continue
scan.
to, yeah.
Continue to, and we have. And so, yes, our model works, and as you know, we've gotten many millions of dollars back, even just this last year, on our investments in biotech companies that have successfully moved forward. Yes, as I mentioned earlier, I mean, most of those 35 trials that you mentioned are phase 2 trials, and about half of them are repurposing trials.
Mm-hmm.
So 2 things. One is the 95% of drug development programs fail. It's been said that, people working in the pharmaceutical industry can spend their entire lives never working on a drug that comes to market, but it doesn't reduce the excitement of the science for people, and it doesn't mean that we don't learn from every single clinical trial that's done. So we're always standing on the shoulders of giants. I think that I can't predict exactly what is going to come next, but I did mention out of our portfolio that I'm very excited about the announcements that might be made in July at the international meetings that will be represented by our investigators in reporting their results on clinical trials that we funded that are due to report out this summer. We already have, positive results on our repurposing trials. We published a very important white paper that resulted from an advisory panel of leading experts on the value of repurposing. It was widely recognized and was accepted for publication and published in a leading neurology journal, International Neurology Journal. And I think our mandate now is to figure out the commercialization strategies for these repurposing trials. How do we move forward? When we show that a drug like Riluzole, which is an ALS drug that I've used in my practice and I'm quite frankly quite excited about, we've seen the results from the trials, the paper's going to be published, but we have a commercial strategy because that's a space where the foundation can play a critical role, because big pharma generally, the revenues from these repurposed drugs are not $10 billion, but they're clinically important and they're important for patients. And so I think it's a real niche for the philanthropy to participate in not only supporting the clinical trials in the phase 2 of repurposed drugs, but to also have that commercialization strategy and helping investigators to move these drugs forward so that the clinical trials don't just become a piece of paper or a report in the medical literature, but they actually become available for patient access one day. And we could have a whole conversation about the challenges in repurposing, but I won't go there. So I'm really excited about our portfolio, Mark, and with the support of all our donors and so on, we're seeing a lot of clinical trial opportunities, a lot of drug development, a lot of innovation. There's more and more companies and academic investigators that are looking at non-amyloid approaches, and I think ADDF has a lot of opportunities to continue to participate in the development of new drugs for Alzheimer's disease.
Yeah, and Howard, that really kind of slides us into, a last takeaway point for everybody that's listening today. One, we have the ability to diagnose with a blood test. Two, we have the ability now, because of a PET scan, to see if a drug's really having effect. And three, now, and Howard, I know this because you and I have talked about each other, Howard and I hope that the biggest takeaway from this moment with Adu and approval is the attraction of more capital to this niche of life sciences, and more capital specifically. I remember Mike Milken was talking with Howard and I a year or two ago at his conference, and he said about the Alzheimer's space, it's not that there isn't enough capital to solve this problem, it's just the capital's aimed at the wrong places. And I know we have some of our donors on today that work in their day job in those capital markets and pinpointing this capital now at these phase 2s, at these combined therapy potential phase 2s, many of which are repurposed drugs, could really accelerate us here. And I know I speak for Howard and myself when I say our greatest hope from this, everything you've heard about Adu one way or the other, that attraction of more capital to this market, that the ADDF, and this is why folks like Gates and Bezos support us, because we know how to pinpoint that capital. So a lot of excitement. I want to do a couple of thank yous before we sign off here. One, I want to thank our staff, our communications and our events staff for putting this together today. As soon as the news came out, they were like, hey, we got to get to our donors, and this has been a wonderful thing to do. Howard, you're brilliant. It's a pleasure. It's a pleasure and an honor, Howard. And there's such excitement here. I can honestly say to everybody, two things about Howard. His true north is, can we get treatments to his patients? And I can also say that piece where he says how excited he is, he lives and breathes that in the office. And it's not new. He's been saying for the last 3 or 4 years that this moment is upon us and is happening. And Howard, you're going to get last word, so don't worry. Howard, I want to thank the lauders. And folks, I really mean this. If you ever need an example of philanthropy making a difference, ADDF in this moment, and hope being on the horizon and hope being here now, is absolute proof of that. And I want to thank each and every one of you that's on today. You were as exciting as it is to get a first FDA approval in 17 or 18 years. And please, you have to feel part of that today after listening to Howard. But what's more exciting is that your participation in philanthropy in Alzheimer's through the ADDF is only going to be speeding what now is to come. And that's even more exciting to do. So thank you all. Howard, last word.
Yeah, I don't think of myself as a genius, but I do want to express my gratitude to everyone for making our work possible, because I know that without your support, I wouldn't be here. And I want to reiterate what Mark said, which is I'm very grateful to Ronald and Leonard for structuring this whole thing from the very outset. And they were really the true North Star. And I've been just trying to execute their vision all these
Amen.
years. So I'm very grateful to everyone, and grateful to be here. Thank you very much.
Good. Have a great day, everybody. Take care and thank you. And we'll be in touch. More to come.